Yeah. Good day, everyone, and welcome to our second webinar hosted by the MNS and in collaboration with the Brain Organoid and System Neuroscience Journal. My name is Nermeen Zakaria, and I'm delighted to be your moderator for today's session. We are thrilled to have you and to continue our series, building on the success of our first webinar. We are excited to have you all here today with us. And whether you are joining us today for the first time or you have been with us before, we are appreciating your participation and enthusiasm. So please feel free to share the link for the meeting with everyone you think will be interested. Before we begin today's webinar, we are happy to have with us Christina Dalla, the head of the MNS organization, to give us a short brief about the activities in the MNS. So, Christina, how are you?
Thank you very much. And hello, everybody. I would like to welcome you today at this second webinar, which I'm sure is going to be as amazing as the first one. And I'm sure you know, but it's good for all of us to remember that the Mediterranean Neuroscience Society is a society for all Mediterranean countries, and not only. Of course, everyone is welcome. And we are. Our aim is to host meetings for all neuroscientists that they are interested in the area. The next neuroscience meeting of MNS will be at Crete in 2025, next year in June, and you are all welcome. You can also visit our website and social media to stay informed about it. And also we are trying, of course, to foster collaborations and educational activities like this one. And I would like to thank very much Dr. Nermeen Zakaria for the organization. And of course, Dr. Luisa Pinto, who is our speaker today. And she's also the Vice Secretary General of the Board of the Mediterranean Neuroscience Society. So please, I am also very excited to listen to this talk.
Yeah, of course. Yeah. So today's webinar is titled Impact of Adult Hippocampal Cytogenesis for Brain Circuitry and Behavior in Depression. And we are honored to have with us Dr. Luisa Pinto. She's a renowned expert in neuroscience. Dr. Luisa has graduated in biochemistry by the University of Coimbra and performed an internship at the Karolinska Institute in Sweden on therapeutic tools for Parkinson's disease. She obtained a PhD in neurosciences in Minho University in Germany in 2008, and she conducted postdoctoral research in the lab of Professor Nuno Souza at the Life and Health Sciences Research Institute, University of Minho in Portugal. She is currently a principal investigator with tenure and team coordinator of the brain circuits and neuron glia adaptations, focusing on novel therapeutics for depression and neurodegenerative disorders. Luisa is also an invited assistant professor at the School of Medicine at the same University of Minho in Portugal, and member of the direction of ICVS as elected representative of the team coordinators of the Institute. Luisa, interestingly, is a COO of a startup company, the BNML Behavioral and Molecular Lab, and co-founder member of the European College of Neuropsychopharmacology Thematic Network on Resilience. We are very happy to have you with us today, Luisa. And before we get started, I just would like to hint some few items that the webinar is being recorded. And the recording will be made available to you afterwards on the website of the MNS. This session will take about 50 minutes, and after that, please feel free to ask. And if you have any questions, you can raise or use the raise button, or just type it in the chat box. And for any technical issues, feel free to reach out through the chat. So, without any further ado, please join us in welcoming Luisa, Dr. Luisa Pinto. We are very, very happy to have you with us today. And
Thank you.
Yeah.
Thank you. Thank you, Christina. Thank you, Nermeen, for the introduction, and of course for hosting these webinars that I think are very, very helpful to all the society to know what we are doing. And I try to do it a little bit educational as well, so that I can show you not only our results, and I will now start sharing, share our results, but that you also learn about the topic a little bit further, okay?
Yeah,
So
sure.
I will now switch to the presentation full screen. Is that okay? Good. So, what
Can you please do it?
Luisa, excuse me, it's not on
Sorry.
full screen. It's
No?
we cannot see the full screen. We see it in the other
Still?
mode. Still, it happens sometimes with Google Meet. I think you need to share your whole screen and not just.
Maybe if you can
PowerPoint.
start the slideshow, maybe it will. I don't know.
I think she has already, but it's not working.
Yes, it's not working because it's already full for me.
Yes. Okay. So if you stop sharing and you share again your whole screen, and not just
Not just the PowerPoint, not just the PowerPoint, the whole screen.
No, try.
Yeah.
But it's not showing up. You are seeing what I'm doing, right?
Yes.
Yes, that's, but stop sharing first. Stop sharing first, and then repeat it.
Let me see. If I do it like this, it also probably can see it, no? You see it nicely like this? It works or not?
But still, it's not full
well,
screen.
but
We see it's not full screen, Luisa. You need to stop sharing. Stop sharing once.
I try.
Now you stop sharing. Now choose the whole screen. You have two screens. That's why it happens, correct?
Yeah. Let me see if now it works. I did it previously, but don't know it. And now?
Put it full screen.
It is?
No, we see. You see, you go on the top. You go on the top of your screen, and then you see. Oh no! Stay as you are where. Stay as you were with the full screen, and on the top, in the middle, either define the monitor, change it. Change it. Yes.
Fine. Okay, it works.
Very good.
Thank you. Sorry.
It's good that Ioannis can speak Portuguese.
Yes, we can speak Portuguese.
Yes.
Good. So I will now start presenting. Sorry for this trouble in the beginning. And as I was mentioning, I will try to tell you a little bit more about the process of brain plasticity in the brain, and then go a little bit further into the work of my team. And I would like to start by telling you one sentence from Stephen Hawking, where he mentioned that intelligence is the ability to adapt to change. And in reality, we know that we are all able to adapt to environmental challenges every day. And this is actually a lot too because of processes of plasticity that we have in our brain. And there are many processes of adult brain plasticity that include epigenetic modifications, functional plasticity, structural alterations of the cells. But there's also one process that will be the one that we will be focusing today that we call adult cytogenesis, meaning the generation of new neurons and new glial cells in the brain at adult ages. And now we will focus on this process and try to understand what it is, how we discover it, and what is the relevance of this process. Okay, so like this, we go back to Joseph Altman's studies many years ago, where he did a very simple experiments by labeling or injecting bromodeoxyuridine, called BRDU, into animals. That is a thymidine analog, and label cells that are proliferating. And what Joseph Altman saw, and this is a very simple experiment, what he saw in the adult brain was that there are in fact cells that are able to proliferate. And this brought us to the existence of stem cells in the adult brain, meaning that we have the capability of generating new cells. Although this was a finding already from many years ago, as you can see, only in 2018, we were able to develop the technology to imaging these stem cells and to really see that in the adult brain, we have these proliferating cells able to divide and able to generate new neurons and new glial cells. This was done in mice as the experiments of Joseph Haltman. And of course, what is really relevant to understand is if this process of generating new cells is very specific to rodents or is actually widespread in other living beings. And I show you here one study from Bali Quiff, where she studied this process of proliferation and the generation of new cells in several living beings, including fish, birds, reptiles, but also primates. And what she could see is that in all these species, we could see that adult neurogenesis exists. And this brings us to humans. So what happens in humans? This is a process that is relevant also in humans. The first study that we have claiming that, in fact, we have stem cells in the adult brain of humans comes in 1998 with Erickson lab and Fred Gage lab, where it showed that, in fact, there are these proliferating cells labeled with BRDU, the marker that I explained you earlier, in the adult brain of rodents, of humans. So this told us at that time, it was very exciting paper telling us that, in fact, also in humans, we have the existence of cell cytogenesis, what might tell us that this is very important for some functions of our brain. However, many other studies came, and some quite recently in 2018 and 2019, that brought us some confounding results with some labs, as for example, Boldrini in 2018, showing very clearly that we have markers of cell cytogenesis and that we have the generation of new cells in humans, while there were other labs claiming that there's no adult neurogenesis in the human brain. And I will show you one of the studies from Maria Lauren Martins in Spain in 2019. As you can appreciate, this was a debate that was really recent, where she actually, this paper came and helped us to come to the conclusion that adult neurogenesis exists in humans. And for example, what she observed was that in patients with Alzheimer's disease, we have a decrease of this process of cell cytogenesis. Again, telling us that this process might be very relevant in healthy contexts, but also in the context of neurodegenerative diseases, for example. And now let's jump to understand where this process happens and how it is relevant and why. So there are, as you can appreciate here with the numbers, there are several articles where show proliferation and generation of cells in different regions of the brain. However, what we can tell by now still is that there are two consensual cytogenic niches here in green and in yellow. And these are the hippocampal dentaginous and the subependymal zone of the adult brain, where we can find stem cells. And this is a particular regions where there are many papers now in rodents showing that they are relevant and the generation of new neurons there. And today, I will be focusing in one of these consensual brain regions, that is the hippocampal dentagyrus. And I will explain you how this process happens. That is quite similar to what happens in the subependymal zone, although, of course, there are some particularities in for example, in the genes being expressed during this process. And what happens here is that we have stem cells with this radii-like shape that divide into progenitor cells, that is called the transient amplified progenitors, that will then divide into immature neurons. And in a process that takes up to 4 to 8 weeks, we have a process of maturation of these neurons, and the fully mature and integrated neuron in the circuit only happens between 4 to 8 weeks after its generation. However, as you can appreciate in this left side of the slide, not only neurons are being generated in the adult hippocampus, but also glial cells and specifically astrocytes are being generated in the hippocampus, although at a lower percentage. About 15% of all the cells being generated correspond to newborn astrocytes. However, we still don't know much about this process, how it happens, and what is the functional relevance of these newborn cells. Let's go deeper now into how can we study this process, right? So we know the cell types, the process to generate a new neuron and a new astrocyte. And what we also need to know to be able to study this process is markers that are expressed within each cell type of the process of cell cytogenesis. And I will not specify them, but as you can appreciate here in this scheme, there are specific markers for stem cells, for the progenitor cells. There are specific markers for the newborn cells, for example, the cortin, as well as specific markers for the astrocytes, the mature astrocytes, like eccentroids beta. This is very important for the experiments that I will show you, because with these specific markers, we can identify the cells. And this was how we were able also in the human brain to show that we have cell cytogenesis happening, because using these markers. Good. What is the function of this adult brain plasticity? And focusing on the generation of new cells, what has been shown in the literature is that cell cytogenesis is very relevant mostly for cognitive performance. And these specific types of cognitive performance, such as spatial reference memory, behavior flexibility, or pattern separation. But there are also reports that show that cell cytogenesis is very relevant for the modulation of emotional behavior, as for example, depressive-like, anhedonic behavior, or anxiety-like behavior. And what my lab has been focusing is specifically how relevant is adult cytogenesis is for function, also considering the dorsoventral dichotomy that we find in the hippocampus. And what do I mean with dichotomy? What is also very special in the hippocampal depre gyrus is that we can kind of divide it in different regions, and considering the dorsal pole and its connections to, for example, cortical regions or to the striatum, it is shown that it's very relevant for the modulation of cognitive performance. However, if we go to the ventral part of the depre gyrus, and its connections to other brain regions, such as amygdala, brain nucleus of the stria terminalis for others, it is shown that it's very relevant for the modulation of emotional behavior. So when studying the function of this process in the hippocampus, we should take in consideration this differential connectivity between the dorsal part and the ventral part to study the impact on behavior. So now I will jump to what we have been doing in the last few years in our lab to try to understand what is the function of cell cytogenesis in the healthy brain, but also in the context of neuropsychiatric disorders, more specifically depression. And for that, I have been using a genetic cytogenesis ablation model that I will now present to you. This is a rat model, the glial fibrillary acidic protein thymidine kinase rat, in which neural stem cells that can be labeled with GFAP marker, after administration with ganciclovir, will be killed. If we kill the neural stem cells, we will ablate the generation or impede the generation of new neurons or new glial cells to happen in the adult brain. And this is exactly what we aim to do. If we inhibit or ablate cell cytogenesis in the adult brain, what is the impact to the function of the brain and for behavior? So this is the timeline that we use. We gave ganciclovir during 2 weeks to the animal, and then studied the brain of these animals at 2 time points that we call short-term analysis, to try to understand what is the impact of immature neurons or cells being generated, and 4 weeks after ablation that we call long-term analysis, to study what would be the impact of ablating mature newborn cells. So for sure, what we need first was to try to understand if the model is in fact valid, and we have to show that our model induces, in fact, ablation of the generation of new cells. As you can see with the marker of double cortin for immature neurons, in a wild type, you beautifully see these cells present, but in our transgenic animals, we hardly can see the presence of a double cortin positive cells. These go that short term. And when we go to the long-term analysis, we also hardly see a presence of a new neuron or a new glial cell in this brain. So we reduce highly the generation of new cells, validating this transgenic model for our purpose. So let's see what is the impact of ablating these cells into behavior function. So we did a battery of tests, and I will explain you what are these tests. And at the short-term analysis, we wanted to understand, to remind you, what is the impact of ablating these immature newborn cells in the brain. We performed a battery of tests to evaluate emotional and cognitive performance. To evaluate an hedonic behavior, we used the sucrose consumption test, in which the animals have to choose between drinking water or drinking sucrose, water with sucrose. And in general, they prefer to eat the sweet water, the sucrose, and the higher preference shows their hedonic behavior. If they have a decreased preference for sucrose, it tells us that the animal has an hedonic behavior. The other test that you see here also measures hedonic behavior, and is called the sweet drug test. The difference here is that the animal has to choose not between water, but regular foods or Cheerios that are sweet pellets, okay? And again, if they have a lower preference for the sweet pellets, it means that they have an hedonic behavior. What we observed at this time point was that, in fact, we don't have any change in the hedonic behavior of the animals. Then we also evaluate anxiety performance. And as you can appreciate here, what happened? Sorry, I go back. Okay, so what happened in the anxiety-like behavior test is a novelty suppressed feeding test, is that we put in an arena, we put a pellet in the center of the arena after food depriving the animals. And the animals that are food deprived have to go to the center of the arena and bite the pellets. As the center of the arena is an anxiogenic environment for the animal, as the longest time that it takes to reach the pellet means that they are more anxious because they are avoiding to go to the center of the arena. And what we observed specifically this time point was that animals had increased, transgenic animals with ablation of cytogenesis present increased anxiety-like performance. And this was the only behavioral changes that we observed at this time point. The picture changes when we go to a later time point that we call the long-term analysis, where we expect to have mature newborn cells. And as you can appreciate, when we look to the tests for hedonic behavior, we have in these transgenic animals the presence of an hedonic behavior. They are, again, presence of anxiety-like behavior. But when we look to cognitive performance, what we could also observe using these tests in specific, the contextual fear conditioning test, that I will also explain you because I think this type of webinars is needed for you to also understand the details of this research. And what happens in this test is that we put the animal in a certain context, we give them a light and a shock. And the animal, of course, reacts to this shock. And the reaction is measured with freezing, percentage of freezing of the animal. In the day after, what we do is we put the animal back in the same context where it had previously a shock. But now we don't give a shock. Anyway, the animal will freeze because it will associate the context to the shock that he got in the previous day. What happens is that when we ablate cytogenesis in these animals, the animals freeze less. And this means that they have impaired hippocampal-dependent contextual memory if we don't have cell cytogenesis happening. We also performed electrophysiological recordings in the brain of these animals, and basically we measure how different brain regions communicate with each other after ablating cell cytogenesis. And summing up these results, what we observed between the dorsal hippocampus and the medial prefrontal cortex, what we see is a decreased communication, or that we call impaired coherence, between these two regions, specifically in the theta band. Showing again that although what happens is that we have impaired communications between regions that are mainly responsible for these cognitive performance impairments that we have in these animals. Summing up these results, so as a brief summary of what we observed, all this study was done in males, and what we observed was that, in fact, if we ablate cell cytogenesis, we showed that the immature cells are relevant to induce any anxiety-like behavior in animals, but at the long term, what we see is a broad spectrum of changes, including anhedonic behavior, anxiety-like behavior, cognitive rigidity, and mnesic impairments. And this was the first study that, in fact, showed that time of the process of generating a new cell in the adult hippocampus is very relevant for functional outcomes, meaning that early proliferation and early survival is very relevant for the modulation of anxiety-like behavior, while decreased neurogenesis and gliogenesis impacts not only anxiety behavior, but also cognitive performance and hedonic behavior. As I told you, all the study was done in males, and you know that there are differences when we think about the context, for example, of neuropsychiatric disorders. We have females that either report, or the levels of reported depression is higher, or double, even the double in females. So it's also very relevant to understand if the cell cytogenesis leads to the same emotional alterations also in females. So we started also to do the same analysis within this transgenic animal model that I presented to you, but now specifically in females, and compare the process of what is happening in males. And I will focus here first in the corticosteroid measurements that we did in these animals. And what we observed in these transgenic females was that the levels of corticosteroid was constantly high, both in morning and night, while we had a normal circadian fluctuations of corticosteroid levels in the male transgenic animals. And this for us was curious and led us to think of a stress response, if these females would be responding to a stress in the same way that males. So we decided to do the dexamethasone suppression test after an acute stress. And what we did, we administered dexamethasone to the animals, waited 90 minutes and did a restrained stress during 60 minutes, and then collected blood to the animal throughout time at 10, 30, and 60 minutes after the stress. And what we observed in the transgenic females, here represented in red, was that the levels of corticosteroid were increasing with time, what was not seen in males. And actually, what we saw in males was the opposite. We saw a decrease in the response after an acute stressor with time. This showed us that, in fact, we have a sex-differentiated response to an acute stressor in males and females that might be correlated with this hypercorticosteroidemia that we see in the female transgenic animals. So we went a little bit further and tried to understand in which regions we might be having an impact at the cellular level, and we analyzed the morphology of cells, and I would focus in and direct you to these results here. And what we observed was that in the basolateral amygdala, we saw hypertrophy of the neurons, specifically in females, and we didn't see these results in males. And looking into the behavior, we compared again females and males, doing the same battery of tests that I presented earlier to not messing up with more behavioral tests. So again, sucrose preference test to measure hedonic behavior. We didn't see any differences in males or females. When we looked to the novelty suppressed feeding test to measure anxiety, we saw in males a clear anxiety behavior that I showed you before. And in females, we also saw increased anxiety levels, specifically in non-proestrous space. What was also very relevant was that we had also deficits in sociability in this test, so that decreased interaction in the sociability tests, specifically in females. So as we have these differences in behavior performance and hypercorticosteronemia, we decided to go deeper and also understand if the connectivity of the brain of these females would be changed also after cell cytogenesis. And what do I mean with connectivity? So what I decided to analyze was after ablating cell cytogenesis, let's look into the inputs to the connections to the dentate gyros from the different regions, from all the regions of the brain that are connecting to the hippocampus. And we did this using a viral-mediated retrograde tracing that we call rabies virus. And we looked into local connections and extra-hippocampal connections. And summing up the results, basically what we saw was in general decrease of the inputs to the hippocampus, more striking from the CA3. And regarding extra-hippocampal connections, we have a high decrease in the number of connections coming from the lateral interrenal cortex. And I will give you our model to sum up these results that can be a little bit confusing, but what we believe that is happening in females after ablating cell cytogenesis. So as you may know, environmental cues are received through the lateral interrenal cortex, and they are sent through the perforant pathway to the hippocampus. When this message comes into the hippocampus, in these transgenic animals, we don't have newborn cells being generated. And the outputs that are sent lead to hypertrophy of neurons in the basolateral amygdala that directly or indirectly through the brain nucleus of the stria terminalis lead to increased anxiety in these females. Downstream of the basolateral amygdala, we will have a change in the communication between the pituitary and adrenal glands, leading to hypercorticosteronemia, constant high levels of corticosteroid that will go back to the hippocampus and lead as a response to high levels of glucocorticoid receptor and its chaperone 90. So this is the model that we have showed up for females that is not actually happening in males. So as you can appreciate here, it's very important then when we look to these processes of cell cytogenesis that we evaluate both sexes as well, as the mechanistic changes are different, but also the behavioral responses and circuitry alterations are also different. So now I will go to the second part of this talk, and all this first part was actually manipulating cell cytogenesis in a healthy animal. And as you saw, we have impact in emotional, but also in cognitive performance of the animals. And the behaviors that we saw alter are actually very characteristic of depressive patients or depressive animal models. And the other part of my lab has been focusing throughout the years in trying to understand the relevance of cell cytogenesis in the context of depression. And of course, I will not go through all these studies that are quite old, let's say, but I will sum up what we have been showing in the first years of this research. And basically, we show that adult cytogenesis is very relevant for the installation of depressive-like traits, but also for remission from behavioral impairments. And they also very important to underlie the efficacy of antidepressants. And now I will just show you a piece of data that is very relevant for what I've been showing. In 2015, we decided to do implementing or perform transcriptomic analysis in an animal model of stress-induced depression that is called the unpredictable chronic mild stress. in which we expose the animals to mild stressors in an unpredictable manner during 6 weeks in a row. After these 6 weeks, we induce to the animals depressive-like behavior, and we have emotional and cognitive impairments in the animals. And when we treat these animals with different classes of antidepressants, as you can see here, we see recovery of these behavioral impairments. So we decided to isolate the dentagitis of these animals and do transcriptomic analysis. And after doing this transcriptomic analysis, we looked to enrichment or gene expression enrichment in the different cell types, neurons, astrocytes, and oligodendrocytes in each of the conditions that I explained you earlier. So what we wanted, the message we wanted to take out of here was to understand what would be, or which genes would be more altered, and in which cell type the genes are actually expressed. And the surprise came when we looked to our stress animal or depressed-like animal, in which, as expected, neurons were severely altered when compared to controls, but the cell type that had a higher number of genes differentially altered or comparing to control was actually astrocytes. And this, of course, was ringing a bell. And we started focusing and understanding not only the impact of neurogenesis, but also thinking of the generation of new astrocytes in the adult brain. And different pieces of data in our lab showed, in fact, that both cell types might be very relevant. If we have normal levels, we have shown that if we have normal levels of neurogenesis in the adult hippocampus, we are able to confer resilience to a recurrent episode of depression. However, if we have low levels or even an exacerbated levels of new neurons being generated, we confer susceptibility to recurrent episodes of depression. Other set of studies that we performed show that also astrocytic cells are altered by stress, as you can see in gray. In the UCMS animal, we see an increased number of astrocytes being present, and treatment with different antidepressants, namely the serotonin reuptake inhibitor fluoxetine and the tricyclic antidepressant imipramine, were able to reestablish the levels of astrocytes. So not only neurons are actually relevant, astrocytes might also have an important factor here. And in fact, what we have been seeing in the literature is that when we think about cell cytogenesis in the adult brain, most people is biased to the generation of new neurons. And this led to a neurocentric view in the literature, and the generation of new astrocytes has been actually neglected. However, now the studies that I will be showing you will be focusing on this process and trying to show you that this process might be also very relevant in the context of depression. Why astrocytes? Why did we thought that they might be relevant? And there are many functions that are important in this context, and I will mention here few. One of those is very relevant for the regulation of neurogenesis. Is very important for the regulation of blood-brain barrier, but it also modulates behaviors such as cognitive and anxiety-like behavior, and it participates in the tripartite synapse, communicating bidirectionally with neurons, among several other functions, like, for example, releasing neurotransmitters. So these are very relevant cells, but we don't know if newborn astrocytes will have a particular function as well. What we decided to do first was to see if, in fact, astrogliogenesis is altered in this stress-induced animal model of depression. And in red, you can see a decrease in the number of newborn astrocytes being generated in the hippocampus, the gyrus of these stress-induced animals. And only the tricyclic antidepressant imipramine was actually able to reverse these low levels of astrogliogenesis. When we look to the morphology of these newborn astrocytes, you can also appreciate that we have an increased number of processes in the stressed animals and in the fluoxetine-treated animals, but the animals treated with imipramine, this tricyclic antidepressant, were actually similar to the control animals, showing that this particular antidepressant seems to be modulating astrogliogenesis in the brain. So we went a little bit further, and I'm showing you here some genes that we have analyzed that are important for the shift between neurogenesis and gliogenesis in the adult brain, as for example, BMP4 and STAT3, where we can see in the adult hippocampus that imipramine leads to a high increase of expression of these genes, showing us that, in fact, it is able to potentiate the generation of new astrocytes. On the opposite, fluoxetine was not able to increase the levels of these genes. And what is also very interesting in my perspective is that when we think about hippocampal neurogenesis, remember I told you about this dichotomy between the dorsal part and the ventral part. We see that neurogenesis is altered or decreased in the dorsal part and the ventral part, and the antidepressants are able to recover in the dorsal part and in the ventral part of the lentigyros. If we look to gliogenesis, what was very interesting to see was that the antidepressant imipramine specifically was able to recover cell cytogenesis, cell astrogliogenesis, only in the dorsal part of the lentigyros. And why is this relevant? Remember when we thought, I told you that the dorsal part seems to be more related to cognitive performance, while the ventral part is more related to emotional performance. So probably these newborn astrocytes, was my idea, of course, would be very relevant for cognitive performance in the context of depression. How to study this? So the strategy that we used, we go back to the transgenic animal that I presented you earlier, the GFAPTK transgenic animal, where we treated with ganciclovir and we ablate cell cytogenesis. What we did now was to do transplantation of glial-restricted progenitors after ablating cell cytogenesis. And after 4 weeks, we saw the behavior of these animals and see if we were able to recover performance. And we did transplantation of the glial-restricted progenitors, but also injection of the secretome of these cells. The secretome would be everything that is released by these cells, we collect, and then we inject in the hippocampal lentigyros. And as you can appreciate here, what we observed, we injected these animals in the dorsal part of the lentigyros, exactly because of what I told you before. We only saw these differences in the dorsal part in astrogliogenesis. And what we see is that glial-restricted progenitors, that I will call from now on GRPs, and the secretome is able to reverse the anxiety-like behavior of these deficits that we see in these animals, both in the elevated plasmase test, but also in the novelty suppressed feeding test. We did 2 tests to evaluate anxiety-like behavior. To evaluate depressive-like behavior, we use the forced swimming test, in which the animals have to swim in a place that is inescapable for them during 5 minutes, and we measure the percentage of time that the animal is immobile. And what we see in the transgenic animals after ablating cytogenesis is that we have an increased depressive-like behavior. When we transplant with GRPs, we are able to reverse this depressive-like behavior. Interestingly, we were not able to recover cognitive performance in these animals. And as you can appreciate, these are the transplanted cells. And we studied, of course, and you can see in the publication, we studied the cell fate of these cells. And we were able to show that, in fact, the GRPs only give rise to mature astrocytes and few oligodendrocytes. Hardly could see a neuron being generated, showing that, in fact, what we are promoting in this animal is the generation of new astrocytes, and the impact that we have in the recovery of anxiety and depressive-like behavior might be due to the generation of new astrocytes. We then looked if these glial-restricted progenitors also influence the environment meaning if they also increase cell cytogenesis in the surrounding environment. And indeed, this happens. What we see particularly in the ventral dentagyros, we see an increased number of new neurons being generated, and the new astrocytes are also being generated after transplanting these cells. We further decided to analyze the secretome and also the tissue, the dentagyros, and did proteomic analysis to try to understand which pathways we are altered, which genes will be relevant to induce this behavioral recovery that we observed. And not going deeply, but you can appreciate from these pathways that mostly of the impact is on neuron development, glial cell differentiation, regulation of neurogenesis, regulation of gliogenesis. So all these processes related to brain plasticity and cell cytogenesis in the brain, making sense according to the results that we obtained. And to have I would leave you with the model that we have running in our lab. And basically, we want to propose an integrative view where newborn astrocytes will not be neglected anymore in the context of neuropsychiatric disorders, more specifically depression. And in fact, our studies show that not only neurons being promoted by antidepressant treatment, but also the generation of new astrocytes is relevant in the context of depression and is very important for the recovery, especially of emotional impairments that we see in these animals. I would just like to finish with the acknowledgements to collaborators, of course, but especially for my team that are a great team and that performed most of this work. And also thanking the funding for supporting these studies. And thank you for your attention.
Thanks a lot. Thanks a lot, Luisa. This was an amazing talk, indeed.
Thank you.
Okay, so yeah, Christina.
Yes, thank you very much. I knew it would be amazing, but every time I discover new data and new ideas, and I'm really curious to see the future, the future results. I have a short question, but I don't know if other people want to ask before me. Or I can start.
You can start, Christina.
Yes, I was wondering about, do you see any sex differences in between the ventral and the dorsal hippocampus, the effects that you find in the different areas of the hippocampus?
This is something I already thought, but we still didn't look. When we found these differences in the transgenic animal in males and females, we definitely have to look into that. And specifically when we did the transplantation, for example, we did in the dorsal part in males because the results I showed you are in males, where I showed these differences in the antidepressants. Because at that time, I didn't have data from the females to really understand if this will be relevant also for them. But I truly believe that there are differences in proliferation levels. There are differences in the neurogenic levels, not only in the dorsal ventral, but also the suprapyramidal, infrapyramidal layer of the dentagyros. Also in these layers, there are differences. So I believe there will be differences in the dorsal ventral in healthy context, but also after modulation after inducing stress in these animals as well. But I cannot tell you the data because I don't know. But this is something that we got curious because now we decided to do everything in males and females because we have to do it. It's really striking the differences.
Nice. I'm glad. I'm looking forward to it. Great. Thank you.
Thank you. Thank you.
I also have a question if there's no other queuing.
Yeah, please go
Thanks,
ahead.
Luisa. Really, really great presentation. And you're right. As Christina says, even if you know the literature, sometimes when you connect, you understand more things. And that's very important for the education series and activities that MNS has initiated. Because when you see present, you see the view, something you cannot easily see or read in a paper.
Yeah.
So you're telling us that astrogliogenesis and the generation of new astrocytes is also very important for the circuit.
Mm-hmm.
So this is something new, because I'm not a neurogenesis, let's say, expert, but working with you all these years and following a bit the literature, I've seen that people neglect that area. So
Yes.
how you consider the importance of the astrocytes? You think they regulate what happened with the neurogenesis? Do you think that themselves they can kill or let's say control the genesis of neurons, or it is more like controlling the local network?
Yeah.
And how
Yeah.
I know you have tools, so it would be nice to also,
Yeah.
and I know you're also willing to host students from MNS for some
Mm-hmm.
period that they can do stuff and analyze in your lab. So
Sure.
please give us
Sure.
your view and tell us
Yeah.
how the
I
students from
I
different
I will.
countries can join your lab in Portugal.
Sure. Sure. I'll give you your view, my view. So basically, there are three labs that showed so far throughout all these years that new astrocytes in the hippocampus might be being generated. Although even these three labs have contradicting data and are not able to tell how these cells are generated, and there's no clue whatsoever what is the function. Okay, there's no clue. And this is because we don't have tools. Okay, so basically what we were fighting, and I hope I will solve this somehow. But what we were fighting is for the fact that all the viral genetic tools that we have, they will incorporate in stem cells. So therefore, they will modulate both the new neurons and the new astrocytes, right? So they will modulate all the process. But people using these viruses and the transgenic tools, they always say, okay, we are ablating neurogenesis. And this is not true because we are neglecting the other cell type, okay? But we don't know anything about it, right? So we don't know what is the function. And there are several hypotheses, as John was mentioning. So there would be the hypothesis that these newborn astrocytes, they are few and they are there just as kind of quiescent and as to replace, to help the system somehow when they need it, is an hypothesis. Although I don't think it is, because uh, I have now some data that probably show that they are more active, but it could be, there could be a reserve, right? It could be there that just to happen when they are cell dead, that they would become active and help the system, or they would be generated and they would be immediately more at the local circuits or in the connections that are coming into the dentagyrus. They will be helping in the maintenance and the formation of new circuits. So this is other alternative that we have. But we don't know if they would go and help the system only in specific connections, or if it would be just all over and they will be helping the overall circuits coming from the dentagyrus. So we don't know that. What John was mentioning was that having this in clue, and I had getting data showing that astrogliogenesis is important in stress-induced depression. We developed viral tools, completely new viral tools to ablate specifically newborn astrocytes in the hippocampal dentagyrus. And these tools are working very nicely. So this is not published, is data ongoing. And they work nicely. And we see that if we ablate newborn astrocytes, we have an impact on behavior, okay, in specific behaviors. So it tells me that these cells might be really functional and relevant in the circuits, although I cannot tell still specifically which ones. Then we also have a transgenic animal where we can also follow the process of generation of new astrocytes. And this is something we are doing now to study how they are generated, with whom they connect, what is the structure of the cells, electrophysiologically, how they are they similar to the pre-existing astrocytes or not? So basically, all this characterization that does not exist at the moment, we are doing now in our lab. So trying to understand better this process.
Yeah. Thanks a lot, Luisa. I also have a question if there's still time.
Sure.
Yeah, I was wondering about the transcriptomics and the genes affected in the astrocytes. After the treatment, maybe we can get a clue about the effect of these astrocytes in the on cognitive and impaired cognition and behavior if we study about the genes that are actually differently expressed. So maybe it's the metabolic or the signaling processes, or what do you think?
Yes. So we had this characterization in that paper in psychopharmacology. We saw the pathways that would be involved in these genes, let's say. Apart from the characterization of the cell types, we also look into the genes and also saw metabolic pathways changes that are very different between the different classes of antidepressants that we studied. So you can I will not tell you now all the details on the pathways, but we can find there that we have all this characterization done.
Okay, yeah. So if we can get the link to the paper, or if it's open access, we can maybe share it with the audience.
You can ask me. You can also ask me, and I send.
Yeah. Okay. So one last question: what do you think if there's in the future precision medicine for depression between males and females? What do you think about it?
It's not only between males and females, but each individual. So it's even more complicated than that. So there are studies that we are performing here in collaboration with the Clinical Academic Center, and these are mostly based on fMRI with males and females, and evaluating the response to a first treatment. Basically to understand, these are males and females, but we see responses different between each individual. And this is a way that we are trying to put forward to try to understand the response of each individual to a certain treatment. And if there is, we can estimate if that individual will respond positively to these antidepressants or not. So this is type of experiments that we are doing, and definitely the personalized medicine is important because, as you can see, these of course are data from animals, but the different antidepressants are recovering behavior through different cellular mechanisms. Okay, the cellular mechanisms that they're acting are different. It can be that, in fact, some people will respond better to a certain antidepressant because it responds better to the modulation of a certain type of cells, and because the particularity of these patients has specific behavioral alterations and specific cellular alterations that is different from the other one. So this is a way that we definitely have to go, and we are combining this with cellular aspects, but also with fMRI, because I think it's important to also see the response of people immediately after treatment and before treatment. So we are doing both.
So this is very interesting. I think we Ibtihal has one question. If we still have time, Ibtihal, please.
I think she wrote somewhere here.
Are there any promising biomarkers related to adult hippocampal neurogenesis that could be used for early diagnosis of depression and personalized treatment? And how might emerging technologies such as single-cell RNA-seq uncover novel biomarkers related to adult hippocampal neurogenesis?
We don't, unfortunately. We don't have these biomarkers. Although I believe that is an important process, we don't have so far a biomarker strictly related with adult cytogenesis process that would tell us about the possibility of using it as a personalized treatment. What we are doing with John is a different way, but we are studying exosomes from depressed patients and also Alzheimer patients, but we are focusing here today in depressive patients, and studying these exosomes that will bring from the brain the proteins that might be some of them related with the cytogenesis process or newborn cells. So this is something that we might look into, looking into the exosomes that are released from the different cell types from the brain. We might get there. Also, it's not easy, I must confess. I think it's not easy to go into the newborn cells markers or biomarkers, but this is a way that I'm going with John, and I think this can give us also very relevant results into the biomarkers field.
Yeah, so I think this is great, and looking at the feedback, I think it opens the mind for many exciting questions and further investigations. So now it's time to talk about: is there any opportunity for the young candidates, if they can come and see or have some practices, if you are willing to do so, concerning this?
Absolutely. So this is something that we wanted to do within MNS. So it's opening our lives to receive students, either to learn techniques, either to share knowledge with us. So it depends, of course, on the objective that each person might have. Learn a specific technique, learn how to induce depression in an animal, or even discuss their projects, even can be through a Zoom link. I mean, I'm open to that. If they want to have some discussions, scientific discussions, I think it can be nice to share projects, to discuss science, even for if they don't want to come personally. We are open. I'm open to have this interaction with our society and with everyone that wants to join us.
So this is very enlightening indeed. And Christina, maybe this opens a new idea about the way we can collaborate and foster more studies with the younger candidates. What do you think?
Sure, I agree. Yes, of course. We need to find some like a framework, funding, and find a way to do it. But I think it's a good goal for the future. Very important one.
Another, if you allow me to add, because it's something we will discuss in the new councils, in the upcoming general council, as Christina said, we are also thinking of having a mentoring platform or a mentoring committees where we can discuss with students way to analyze or improve their projects, and as well as to give them fund on specific analysis or small things that they can do one and there. That will also kind of a mentoring, which can be through Zoom or Teams, and that will help a lot different students from different countries. So the idea we have discussed with Christina and Luisa and other members in the council, and we will discuss it more in September, because we had to focus a bit on the preparation of the MNS 2025 that will happen in Crete, in Greece, in June 7 to 11, just to remind to everybody, since it's recorded, so it can play later. We had to focus on that, but the idea is also to create, 2-3 different committees on different focuses on neuroscience that we will gather once per month or once per 2 months, can give feedback to any student or anyone that would like to have some feedback on their projects or ideas to analyze or tools that can available or be sent, a virus or something. It depends on the people that they are joining. And this indeed was something we already discussed with some people in the recent Eurogenesis meeting. Luisa can tell more about that. Mm-hmm. Like people volunteer to help on mentoring and
So this is absolutely something that we can discuss for the future, and try to do something more organized, right? So that people can participate on this, but we will certainly be open to do that.
Yeah, okay. Yeah, so this is really a great session, and with lots of insightful information. And thanks a lot. You've added a lot to the session. Thanks, Christina. Thanks, Ioannis, and of course, thanks, Luisa, for the perfect session. And we look forward to having more and more informative sessions like this. Thanks a lot for your time. And anything to add?
No, from my side, just thank you for being here.
Okay, so yeah.
Yes, thank you very much. I also put on the chat the website of the society.
Yeah.
So please join us and follow us on the social media as well.
Okay.
Thank you very much.
Thanks a lot. Thanks a lot. And we look forward to have further meetings soon. And bye-bye for now.
Bye-bye. Thank you.
Bye.
Bye.